So the immune system chapter, and I gotta remember all the chapters because they make new editions and then I never remember which, they switch chapters around or something. And I want to light off. At least low. So the immune system chapter itself is 3 chapters, 14, 15, and 17. 14 is all about, and I'll go over it, Mike, is all about the innate immune system, which my next slide will talk about what that is. Chapter 15 is all about the adaptive immunity, and chapter 17 is all about how can we use everything we know about the immune system and then use it for our own purposes. How can we use our immune system to test people and confirm they have different types of diseases? So it kind of goes over different types of platforms on how we can test, how we can diagnose someone based on immune system reactions. Chapter 16, there's a reason why we skip over it, talks about all the issues that the immune system can have. It's all immune system disorders. I don't lecture on the immune system disorders. If you're going into the different medical fields, I know most of you guys are going nursing. I'm like, you'll talk more about some of the different disorders when you get into the program itself. But immune system disorders are hard to treat because you pretty much have to kill someone's immune system. So I don't lecture on it. However, there is a PowerPoint review and there is a quiz on it, just so you have some basics on what does it mean to have an immune system disorder. So it's its own separate unit. It's not on the next exam. It's just like its own separate short little quiz. I don't think it's due for a couple weeks, but you'll notice it on the schedule. It'll be in the folder in the Blackboard lecture. It'll talk about it. So it's like I skip over chapter 16 lecture on, and it's not part of the next test, but it's its own separate, just take a look at it, get a little bit of background on it. And then there's just the one homework. It's not like a quiz, timed quiz. It's one of those, take as many times as you want. Just to get a little bit basic overview on the immune system. Now, when I break apart the immune system with the 3 chapters, the 14, 15, and 17, there's a reason why I put chapter 14 online. Out of the 3 chapters, it is the easiest one for you guys to tackle and understand on your own, because this class is blended, so part of your lecture is online material. So as I pick and choose what goes on and what online and what do I want to make sure we go through in lecture, in person, chapter 15 is a harder unit to understand and comprehend, especially if you have to do it all on your own and you don't have me drawing stuff and pictures and arrows and stuff like that. So chapter 14 is on your own, meaning there's already a recording for chapter 15 out there to watch for this week. And then there's an online quiz, the normal 5-point quiz that you can take as many times as you want as well with that. So if you saw that on the schedule, maybe you already watched it, maybe you didn't. Because their immune system is broken down into 3 lines of defense. The first 2 lines of defense, the first line itself is anything that just prevents microorganisms from getting in the body. So our skin, our mucous membranes, they're just the barrier. The second line of defense is other things that our body can do to try to get rid of pathogens, but they're not specific responses. They're not specific for a particular virus or specific for a particular bacteria. These are just things that our body knew how to do since the day we were born. Our body had certain cells that could recognize pathogens and eat them. Our body had various chemicals that it could make, processes like inflammation, fever, things like that. Those are just non-specific things that our body can do to try to get rid of the pathogen. So the first 2 lines of defense are called innate immunity. These are things that our body was just born knowing how to do. It didn't have to recognize anything. It didn't have to remember anything. It's just some generic things. And that's chapter 14. So that's kind of some basic ideas that's usually pretty easy to comprehend. It's chapter 15, the adaptive immune system that can recognize very specific invaders that get in the body. It can mount a whole immune system against that one particular invader, and then this is where we get some of those memory cells so that if you get infected with that same specific invader again, our body knows what to do about it. And so this is where we get some of that memory, why we have immunizations. That's all the adaptive immune response. And there are some specific cells and specific processes that all takes place on how to do that, which is what we're going to tackle, is that adaptive immune system. So the innate defenses are those first two lines of defense, kind of again just showing some different parts of it. So the first line, your skin, your mucous membranes. Your internal defenses, you have phagocytes, you have things called natural killer cells, you have inflammation, different types of antimicrobial proteins, and fevers. Those are all things that our body just knew how to do. It's the adaptive immune system, chapter 15, that we're going to talk about in this lecture. And there are two branches of the adaptive immune system. As I talk about it, I recommend writing it down. And as I talk about it, I'm going to add to it. So our adaptive immunity, like it's adaptive immune system, has two branches to it. We have a humoral branch and cell-mediated, also called cellular. Depends on the book. Now, they do each have their own types of cells that are involved in the two different branches. And my goal today, we'll see timing-wise, is to get through just the humoral branch and what's happening there. On Thursday, we'll talk more about the cell-mediated and then how the two branches can start to relate to each other. But they each have their own types of cell, B cells and T cells, which I'll talk more about coming up. So the PowerPoint is going to go over just, what is adaptive immunity? What are some terms when we talk about adaptive immunity? Where does adaptive immunity happen? What are the main players? What are antigens? What are B cells? What are antibodies? What are T cells? What are cytokines? Some abbreviation things. And then what's all happening in the cell-mediated immunity? What's all happening in the humoral immunity? How can they interact with each other? And then how do we acquire immunity? Like, how do we become immune to different things? So we're going to start first with some terms on what is adaptive immunity. So adaptive immunity means it has the body's ability to recognize and defend itself against distinct invaders, meaning these are very specific invaders. And or any of the products that they make. Because there are some bacteria that make toxins. So our body can recognize not only the bacteria, but it can also recognize the toxin. Use some different colors just because. I'm going to erase these after I write them up, but just to get rid of stuff up here. But there are some terms and attributes when we're talking about our adaptive immunity. The very first one is the specificity. It's the fact that our immune system can tailor reactions to specific invaders. So let's see. It's the immune response against specific invaders. Like very specific invaders. Like our body can recognize pretty much anything that could ever get into our body. And if you get a splinter, our body can recognize it and anything that might be on it. If you get bitten by some type of venomous snake or spider, our body would recognize exactly that venom and different from other venom. Our body can recognize all those very specific things. 2, the inducibility. Those specific invaders will induce or activate cells in our immune system. And I'll talk about that in just a second. So the specific invaders induce / activate cells of the this means that the cells that we have in our immune system are not at war if there's not a foreign object in the body. So our cells are dormant. They're looking for invaders. They're patrolling the body as they circulate around the body. But they're not actually doing anything yet until the particular foreign object that it recognizes is in the body. So they kind of just stay, our cells, our adaptive immune cells, stay dormant until they recognize the thing that they're supposed to recognize, whatever it might be that they're supposed to recognize. Then they become active. So when that foreign object, when that very specific invader gets in the body, then it will activate or induce the cells of our immune system. And then once activated, it goes into what's known as clonality. So once activated, that T cell or that B cell that became activated starts to make clones of itself. So once activated, we'll make clones. An example of why this happens. So we have cells in our body that can recognize COVID, because I'll just use that as an example, but it could be any virus, it could be literally anything that gets in the body. But we've got a cell that can recognize COVID. Once it gets in the body, that cell, once it recognized it, now becomes activated, and it needs to fight a war. Well, the best defense when you're fighting a war is to have the biggest army. And so that's what it's doing. It's, one cell can't attack and get rid of every COVID virus that's in the body. Instead, it's going to make clones of itself. It's going to make a whole bunch of itself, and then that huge army can now go and attack that particular invader, that particular virus, COVID. And so once activated, it's going to make clones of itself. It's just building itself an army of cells that recognize that one very specific invader. Now, our adaptive immune system also has what's known as unresponsiveness to self, which means it could recognize normal body cells and ignore them. So the job is to recognize foreign objects and to know the difference between a foreign object and one of our own particular cells in the body. And again, that chapter 16, immune system disorders. There's lots of different types of immune system disorders, but there's a whole group of immune system disorders where this is not working. And the body will recognize our own cells as foreign and then mount an immune response against it, which is never good when your body tries to fight your own body. And then the last, the memory, is that it will have a faster response with every subsequent, use a big word, infection. Meaning every other time after that, if you get that same particular invader in the body, it hasn't changed at all. It's the exact same thing in the body. We have memory cells that remember what to do. And once you learned how to do something, it's a whole lot faster to do it the second time. Because 1, 2, and 3, recognizing specific invaders, getting activated against them, mounting a whole war, making this huge army, can take 2 weeks to really get a full army response. That sometimes, is when you start to get real sick. But if you now remember how to do it, it could take only a couple days. And again, hopefully you can mount an army and fight things off fast enough before you ever feel sick. Now, the main players in the adaptive immune system are those B cells and T cells. They are lymphocytes. So there are 5 white blood cells in the body. The neutrophils, if I say it right in order. Neutrophils, lymphocytes, eosinophils, basophils. monocytes. 4 out of the 5 are all part of that second line of defense. So that's part of the recording, chapter 14. The 5th white blood cell, the lymphocyte, is part of the adaptive immune system. And so B lymphocytes or T lymphocytes, we shorten it just because it's a long word, and we just call them B cells and T cells. Where they get their letters, the B and the T, is where these cells mature. They're not active yet, so they haven't gone into the inducing active, but they're mature. They're ready to go find some foreign object. So B cells mature in the bone marrow, that's where they get their B, and T cells mature in the thymus, where it gets its T. Now, we're going to hopefully get through our humoral branch today, and the humoral branch is anything that has to do with B cells. I'm not writing up T cells yet for cell-mediated because there's different kinds of T cells. And B cells is easy, just B cells are B cells. Now, the cell-mediated response has T cells, but there's more than one kind of T cell. But they each have their own different kind of lymphocyte. Now, our adaptive immune system in general, what it tries to do is it recognizes very specific foreign invaders, but pretty much anything that we consider as foreign, our body considers as foreign, is considered what's known as an antigen. So a foreign object, something that our immune system can recognize and try to fight an immune response, is considered an antigen. I'm actually going to draw up my little antigen that's up there. Oops, I don't want green yet. I'm going to use the same colors that's in that big blue blob. Breaking stuff up here. Orange circles. So the whole object itself, just, my random object up here, that big blue blob with the reddish, we're calling it red today, red triangle, orange circle, green square, they do have epitopes on it. So the whole thing is foreign object. Our immune system can recognize that whole thing as a foreign object. However, every foreign object can have multiple different extensions sticking off of it. And our body can recognize the whole thing as foreign, but it can also recognize each little thing that sticks off of it as a foreign object as well. So we have cells that can recognize the whole thing. We have cells that can recognize just the little red triangles. We have cells that can recognize just that orange circle, and we have cells that can recognize just the green square, which means we have cells that can recognize the whole thing or the 3 different parts of it, which means we can target this multiple different ways by multiple cells because we can recognize every little object on the antigen itself. Those little objects that stick out of an antigen are called epitopes. So it's just almost like extra things that our immune system can recognize as foreign. Because epitopes are just small structures on the antigen. Now, different books, different pictures. This big, huge thing is showing that this is a particular antigen. The whole thing's recognized as foreign. But yes, it can have different things on the outside of it that are also the immune system can recognize as foreign objects. Or maybe it recognizes as a normal thing, as a self-normal cell. Again, different picture, whole thing is an antigen, different little random things that stick out on the outside are epitopes. So different things that our immune system can recognize as foreign. Again, we can recognize just about anything, but some of the big things that we'll always recognize as foreign, flagella. I mean, the only cell in the human body that can have a flagella is a sperm, but it is structurally different than a bacterial flagella. So our immune system would recognize flagella as foreign. It would recognize peptidoglycan as foreign. None of our cells have peptidoglycan. That's in the cell wall of a bacteria. We can recognize, I mean, not just bacterial structures, we can recognize different types of viral structures. Some viruses, when we eventually get there, have double-stranded RNA. That's really unique. Nothing in our cell has double-stranded RNA. Any RNA that's ever in our body is always single-strand. And so to have something that's double-stranded RNA, super unique. Our immune system is like, oh, there's nothing like that. We can recognize it. But it doesn't have to just be bacterial. It doesn't just have to be viral. Our immune system can recognize other things as foreign. It can recognize certain foods as foreign. If you are allergic to a certain type of food, it's because there is something on that food that the body recognizes as foreign, and it fights an immune response against it, which is going to cause you to have symptoms of whatever food illness you get when you eat something. Same thing if you're allergic to dust, certain types of antibiotics or drugs. That means there's something on those particular things that the body recognizes as foreign, and it will mount a whole immune response against it, leading to all your symptoms. I'm not going to watch that little video link. Now, so antigens are things that the body can recognize as foreign, but we do break our different types of antigens into 3 groups. So it's exogenous, endogenous, and autoantigens. And exogenous, what is, X versus end at the beginning of a word mean? What does X mean? Or outside. So if you exit, you go outside. Exogenous antigens just means these are antigens on the outside surfaces. Example, a flagella. That's on the outside of a bacteria, and there are certain structures on it that the body would recognize. This has like little green triangles. It could recognize viruses, it could recognize bacteria, anything, any outside structures, as long as they're just not inside of one of our cells. It can recognize anything on the outside. So they're exogenous antigens. They're outside antigens. An endogenous antigen is the opposite. These are inside antigens. And we'll talk more about those on Thursday. But we do have issues. So there are some viruses, well, I guess all viruses. There are viruses that have to get inside of our cells to reproduce. There are some bacteria that can get inside of our cells to reproduce. So if a bacteria or a virus is inside of one of our cells, how does our immune system look at one of our cells and be like, there's something inside of you, I need to kill you? Because although we can look at a nice 2D image that's been sliced in half, our immune system doesn't have that option. It can't just see through our cells and know that there's something foreign in there and that it needs to kill that cell because it's infected. Instead, what does end up happening when there is something foreign inside of one of your cells, virus, bacteria, whatever it is, if you notice on the outside of that virus, there are little blue circles. What happens when a virus or a bacteria gets inside of one of our cells? Some of the antigens that that particular organism has are actually presented on the outside of our cells. So though this is an inside organism, those same little antigens are actually shown on the outside of the cells. It's how our immune system know that it's infected. Those little blue circles should not normally be there. Instead, they're there because that cell is infected. And so it's literally the red flag that's like, there's something inside of me, I'm not normal, I'm not supposed to have little blue circles. And our immune system can recognize that, and it can kill that cell. So it's an inside antigen that's presented on the outside. Autoantigens. The word auto means self. If you drive an automatic car, it shifts by itself. Auto means self, and so autoantigens are self-antigens. These are things on the outside of our cells that our immune system should recognize as normal. Should, unless you have immune system disorders. So these are structures on the outside of our normal cells that the immune system should recognize as normal. You have carbohydrates that stick up on the outside of our cells. Our immune system has to be able to recognize those as normal. We have different structures that stick out on our cells that are found in our cell membrane. Again, our immune system should be able to recognize those as normal objects. Normal structures, normal antigens that are supposed to be on the outside of our cells. Again, if you start to have immune system disorders that would recognize those as foreign, it would start to kill our own cells. Now, a couple terms when we're talking about different types of antigens. Again, things that our body should recognize as normal. We have something known as MHC, which stands for Major Histocompatibility Complex. And we have something known as antigen-presenting cells. So starting first with that major histocompatibility complex. So these are antigens on our cells. They are little glycoproteins, little sugar proteins, that are on the outside of our cells. They are found on most cells of vertebrates, and they are not recognized as foreign. So these really are those autoantigens. But there are two kinds. There's major histocompatibility complex type 1 and major histocompatibility complex type 2. Major histocompatibility complex type 1, again, these are self-antigens, things that are normally on our cells. These are found on all nucleated cells. In the body. Now, there's really only one kind of cell that's not nucleated. Does anyone know what it is? At one point it had a nucleus, but then it got rid of it. And so then the cell doesn't look round anymore. Again, I'm not artistic, so we'll see. Red blood cell. Yay! I'm glad you thought it looked like a red blood cell. I picked red also for the reason. Red blood cells, you know, they have that little dip in them, and that's because they don't have a nucleus. They lost the nucleus, so they aren't round anymore. So the only cells in the body that don't have an MHC1 are red blood cells. But we generally just say they're on all the cells in the body. So every single cell in your body has that particular type of antigen on it, a major histocompatibility complex 1 antigen. Now, it's called major histocompatibility complex antigen is because we didn't even know these things existed until we started to do tissue transplants. Anytime we started to put one kind of tissue into another person's body, does that body always accept it? No, because what your MHC1 antigen looks like is different than someone else's MHC1 antigen. So if you're given someone else's cells, those things are going to be recognized as foreign, and your body will fight immune responses against it. And so again, we didn't know these things existed, so that's why they call them major histocompatibility, is because when they do tissue transplants, they try to find antigens that are as similar as possible. Unfortunately, you still have to do an immunosuppressant. You have to suppress someone else's immune system, whoever gets that tissue, because they're going to reject it. That's what the immune system's for. And MHC class 2 are found, and if you've got it printed, you can just draw an arrow. They are found on antigen-presenting cells. So MHC1 on all cells in the body, minus those red blood cells. MHC class 2 are only found on antigen-presenting cells. Now, based on its name, these are cells in the body. A lot of times they're macrophages. Their name implies they're going to eat stuff, and they're going to eat lots of stuff. They can eat foreign objects, foreign bacteria. They can break it down, and then they will present on the outside of that cell those endogenous antigens, they will present those foreign objects to our immune system. Like, hey, I just ate this foreign thing. Look at it. Do something about it, because I'm done eating. So like, it has to wake up our immune system cells. So that's their job, is they're to present foreign objects to different cells in our body to, I erased it, but I'm like, to wake them up or induce them. The top cells in our body that can bring in and then present these different types of antigens, dendrites. And I'm like, now we do have dendrites in our nervous system, completely different kind of cell. They're just called that because they have the long extensions on them, like dendrites do on your axons. But we have dendritic cells, we have macrophages, and B cells can actually present antigens as well. So a couple terms that we've got: different kinds of antigens, and we've got different things on the outside that can present antigens so that we know we have something foreign in the body. Now, we are going to focus on just those B cells and the humoral immune response. So those B cells, they do come from the red bone marrow, but then they also mature in the red bone marrow. And this is where they're going to encounter any foreign objects, as well. They're just hanging out there, sitting in the bone marrow. Where we can find them in action, in the spleen, in the lymph nodes, in something known as mucosal associated lymphoid tissue. I'll spell it. It's good to know. I can't spell today. That's your malt. Now, we do have something known as this malt, and I'll quick talk about it. The mucosa is just like our mucous membranes, anything that lines those internal environments, because they're exposed to the outside environment. Your digestive tract, your respiratory tract, like, we have to be able to recognize these foreign objects that get in there. So your gut, which we even have a whole nother group of gut-associated lymphoid tissue, but your gut and your mucosa, because it's exposed to so many possible foreign objects, we try to concentrate a lot of B and T cells there. And so we've got a lymphoid tissue. It's not its own structure. It's not like a spleen. You can't just remove a whole organ. But it is tissue where you have a large concentration of T and B cells. They're just in areas where it's at a high chance of foreign invaders. And your gut has a ton of them. Your digestive tract is full of different kinds of bacteria. Some are normal, that's great, but not a lot of them are. Again, if you dropped food and then picked it up, just ate stuff off the floor, you're putting lots of foreign objects in the body, and your immune system has to be able to recognize and do stuff about those all the time. And so these lymphoid tissues, they're just tissues with lots of lymphocytes, lots of B and T cells. The blood only has a small number of B cells, but the main job, the main ultimate goal of B cells is to make antibodies. And then the antibodies are going to do something about that foreign object. But I'll talk more about it making antibodies coming up. So again, my recall of my humoral immune branch, the humoral immune branch involves B cells, and ultimately its job is to make antibodies. Now, on a B cell, here's my B cell. It has receptors on it, on the outside. I'm going to draw multiple cells here. I should have drawn them smaller. So I have 3 B cells shown up down here. And each one has a B cell receptor. And the B cell receptors, they look like a big Y. On one particular B cell that I draw up here, it's covered in little red Ys. On another one, it's covered in little orange Ys. When I say covered, I'm drawing 4 or 5. You could have thousands of B cell receptors all over it. And my other third B cell receptor up here has little green Ys. So each B cell only makes one type of B cell receptor, meaning this B cell only has red. This one only has orange. This one only has a green. You're not going to have a B cell multiple colors or multiple different B cell receptors. Every B cell has its own unique B cell receptor. I like to liken them to snowflakes. You know, every snowflake is unique. Every B cell is unique because its B cell receptors are all different from each other. Now, each B cell can make many copies of the same B cell receptor. I have like four red Ys, 5 green Ys, whatever. It makes multiple copies, but they're all the exact same on every particular cell. And each B cell receptor only can recognize one thing. So this green, I color chose them for a reason, this green Y can only recognize on this antigen the little green squares. This orange B cell receptor can only recognize little orange circles. This red B cell receptor can only recognize the little red triangles. They can only recognize one thing. But ultimately, we have enough B cells with enough variety of B cell receptors. We can ultimately recognize millions of different things that can get in the body. You know, I'm showing an example of 3 different B cells that are recognizing 3 different things. But we have millions of different types of B cells. Each one has their own very unique B cell receptor that can recognize its own unique thing that can get in the body. So we have lots of different B cells, each with their own B cell receptor. Zoomed in picture of showing the B cell and the B cell receptor. So again, there's the cell membrane. It's still a cell. It's still a lymphocyte. And that B cell receptor, it looks like a big Y. It has an anchor that helps it anchor down into the cell membrane itself. But at the very top of the Y is where it has, every time I picture this, I picture those blow-up things that like move all around. It's the arms of those things. It's the hands that actually do the recognizing. So they call it the variable region or the antigen binding site because it varies. That this has little red hands, and this would have orange hands, and this would have green hands. It's the hands itself that recognize the different foreign objects and can bind to them. So this one, on its little hands at the top, have a very specific hand that just recognizes that little bluish purple cone. And there'd be a completely different type of hand structured differently that would recognize, squares or circles or whatever it is. It's just the hands that change. Now, how do we activate those B cells to do something? Because our ultimate goal is we need some antibodies to do something about it. So a B cell becomes activated or induced, as we said before, when the antigen, those little hands, binds to the receptors. Now I said and cross-link it. It just means that they're bound to each other. The hands are binding to the antigens. They're binding to that little purple cone, or they're binding to my little red triangles or orange circles or whatever. Once those hands bind to that foreign object, we've now just activated that B cell. That B cell's like, holy cow, I just finally found the thing that I'm supposed to find. It bound to it, it's now become active or induced, and it's going to go through what's known as that clonal selection. It's going to make more of itself because it now needs a big, huge army to fight a war. Once it makes more of itself, it's going to become one of 2 different kinds of cell. So it's going to make more of itself, but then it's going to change a little and become what are known as effector cells, meaning they're going to actually cause an effect. They're going to do something. And one of those types of cells is called a plasma cell. So a B cell, once it's activated, can start to make plasma cells. If it doesn't become a plasma cell, most of them will, Its other option, so it's my smaller arrow just because there's not as many of them, can become a memory cell. But most of them will become plasma cells. I think I've got this picture zoomed in on one of my next slides, so I'll come back to it. Now, as I said before, most of those effector cells become plasma cells. And it's the plasma cell's job to churn out those antibodies. I'm going to always probably shorten it to AB for short. And one plasma cell can churn out 2,000 antibodies per second for about 4 or 5 days, and then it dies. That's hopefully enough antibodies with enough plasma cells, they can go target and get rid of that particular foreign object. So the antibodies are going to circulate around in the blood. You'll find them in the lymph. They're looking for that foreign object. They'll bind to it, target it for destruction, get rid of it. We'll talk about some of those, how it does all that shortly. But it makes lots and lots of antibodies. If it doesn't become a plasma cell, it becomes a memory cell. So we have memory B cells that, when presented with that same object, can do this whole process a whole lot faster. Because activating a B cell, going through that clonal selection, making lots of B cells, then making plasma cells, and the plasma cells churning out a whole bunch of antibodies, again, can take a good 10 to 14 days. Which means you're going to be sick, because you're at that point, 10 to 14 days, the bacteria is growing like crazy, or the virus is growing like crazy, and you're not really doing anything about it yet. However, memory cell, because it now knows how to make these plasma cells to churn out the right antibody, can start churning out huge numbers of antibodies within days instead of weeks. Good, I do have a bigger picture. So up on my top of the picture, I have 3 different B cells. Again, they're different because on the outside of each one is a different colored B cell receptor, just so you know that they're different B cell receptors. There's green receptors, we'll call them purple receptors, and there's a blue receptor. That red object is the foreign thing that's in the body. And if you notice, only 1 out of the 3 can actually recognize that thing. It's the one that's got the purple. I think it's like dark blue. But only the purple B cell receptors recognize that foreign object. These things, this blue, the light blue and the green, recognize something else. Once it binds to that foreign object, once it binds to that antigen, again, it's only the hands that are binding, it's now become activated and it goes through that clonal selection. It starts to make more of itself. And each of the new clones has that same kind of B cell receptor on it. Because again, it's making exact copies of itself. It's fighting a war. Now, it's not just going to make 2, it's going to make thousands, but easier just to draw 2. Once we have a lot of these clones, they then become the effector cells. They're going to become a plasma cell, and most become plasma cells. In the book picture, 3 out of 4. Most become plasma cells, and the plasma cell's job is to secrete out antibodies. If it doesn't become a plasma cell, it becomes a memory B cell. Again, it still has that same receptor on the outside, so that it knows how to recognize those little red things that are up there. Now, looking at the pictures of a B cell receptor on these cells and the antibodies that are made, what do you notice about them? What an antibody looks like compared to what a B cell receptor looks like. Anyone? Do they look different? B cell receptor, antibody? Or do they look the same? They look the same. They are. An antibody really is a B cell receptor that's like free, off doing its own thing, not stuck to an actual B cell itself. And so it should look the same. It's still a Y. It still has those two hands, and it still has those hands that could recognize, and only on the hands that recognize that foreign object. So when these things circulate around, those hands are looking for that red foreign object or whatever that foreign object is that got in the body. So the antibodies are secreted truly look exactly identical to the original B cell receptor that got activated. Now, what is an antibody and how does it work? So antibodies, again, they look like a big Y, and it's only at the very, very top, the hands, that differ really between them, because it's differing based on what it can recognize and attach to. Antibodies are proteins. So people that are sick, and you guys that are going into nursing, it's why you have to preach to your patients. You have to eat protein when you are sick. Antibodies are made of protein. They are protein objects, which means if you're not getting any protein in your diet, your immune system isn't going to be working at full capacity. So when you're making antibodies, you're making protein objects. So you do need a good protein diet. They are made from plasma cells. They have antigen binding sites, the hands, that recognize the antigen, and they are identical to the B cell receptor. They need to be identical because they are going to target that identical thing that activated the B cell in the first place. Now we break these big Ys, these big antibodies, into two regions, variable regions and constant regions. The variable regions are at the tops, they're the hands. The constant regions are the body. Because they're not changing between different things, different objects that they need to recognize. It's just those hands that are changing. So the variable regions are at the very, very top where you have the antigen binding sites. And the constant region, on here is like all the gray and the C, that's where the constant, the C stands for constant, is in like the other part that really has no association with that antigen itself. Just like the rest of the antibody, but it's not doing a lot. Now, its function, as I said, we want to churn out antibodies, and the antibody's job is to get rid of whatever foreign object got in the body. Its job is to bind to antigens. And what it really is going to target is bacteria, different types of toxins that different things can make, and extracellular viruses. The humoral branch, the B cells, the plasma cells, the antibodies, are really looking for extracellular pathogens. Things that are not inside of our cells. Things that are floating around somewhere in the body. They're just not inside of our cells. They have not hijacked our cells. They're not hiding out in our cells. One of the top things that an antibody's goal is to do is to increase phagocytosis. It wants to try to target that foreign object to get eaten so that we can get rid of it. It can also inactivate different types of pathogens or toxins as well. And the how is my next slide. So how can antibodies do some of this? How can they increase phagocytosis? How can they try to inactivate toxins, try to kill the object that's foreign? So again, I've got all my antibodies up here as red. The very first way that an antibody can work is something known as neutralization. So again, it's only the hands that are attaching to the foreign objects. And the hands can attach to a toxin. They can attach to the outside of a virus, or they can even attach to different kinds of bacteria. And depending where they attach to those different things, means those different things, whether it's a toxin, a virus, different types of bacteria, they can't do what they're supposed to do anymore because they have antibodies stuck to them. If a toxin is toxic to certain types of cells, and that toxin needs to get inside of cells, that little tiny green triangle toxin can't really easily get inside of our cells anymore because it's got these big huge antibodies stuck all over it. It can't sneak in. So it neutralizes that toxin. That toxin can't get inside the cells. It can't do whatever it wants to do. A virus needs to attach to our cells and then get inside of our cells. But if all those little blue receptors that's used to find the right cell are all attached to an antibody, it really doesn't have any receptors anymore, so it can't recognize our cells, and then it can't get inside of our cells. So it inactivates the virus. Same thing with bacteria. If it's covering or attaching to all the little receptors that it has, it can't recognize our cells. It can't attach to our cells. It can't find itself a home, because before bacteria reproduce, they first have to find a home. If it can't do that because it's covered in antibodies, bacteria can't grow. So these antibodies are targeting certain areas to stop toxins from getting in the body, to stop viruses and bacteria from getting inside of our cells or calling themselves a home. So that's one way that bacteria can work. Another, so an antibody does have two hands, which means it can potentially attach to two different bacteria. They're the same kind of bacteria. Again, I can only recognize one thing. But it can attach to two of the same kind of bacteria. And if you get enough antibodies attaching to enough of that foreign object, you can clump them together. Agglutination means clumping. It has the word glue in it because it's like gluing it together. So it's going to clump objects. Why that's beneficial for us is some foreign objects, bacteria, even certain viruses that haven't gotten inside of our cells yet, they're so tiny that our immune system doesn't really see them right away. However, it's a whole lot easier to see if it's now this big clump. It's also, if you're a white blood cell that likes to eat foreign objects, it's a whole lot easier to eat a big clump of something than just one tiny thing. It's a lot more efficient. So it clumps foreign objects together. It can also increase what's known or cause what's known as opsonization. Again, immune system has to have all these big long words. Opsonization just means it's going to increase phagocytosis. Like opsonize means to phagocytize. Different words, same thing. So when it's covered in antibodies, there are some objects that our immune system can't see, and if you can't see it, you can't eat it. Not only it's structured somewhere where you can't see it, some things are almost too slippery, things that have capsules that our immune system, our white blood cells just can't get around that object and eat it. However, if an object that's too slippery or too small is now coated and surrounded by antibodies with these little spikes sticking out all over, it's really easy to be able to eat that. It's not slippery anymore. It's got little spikes sticking out on it. And it's a lot easier to see. So it's just kind of like a big red flag on the outside of that organism. So it increases the chance of getting eaten. There is another way that antibodies can target objects for destruction. It's known as antibody-dependent cell-mediated cytotoxicity. I'm not going to ask you to know what that stands for. The idea is that we have cells in our body called natural killer cells. That's what the NK stands for on there. And the object of a natural killer cell is to kill. And it does it by usually secreting some type of, I don't want to say toxin, but pretty much a toxin that's going to cause holes to form in the bacteria. So what these antibodies do is their hands are going to attach to the bacteria, but the tails are sticking out. Natural killer cells recognize the tail. I mean, it in itself is still an object, and natural killer cells can recognize the tail. And so it's like, oh, hey, there's some tails. They must be attached to something. And it will attach to the tail, and then it will secrete something that causes holes to form in the bacteria, and the bacteria die. So it's a group effort on how to kill these particular organisms. The antibodies are just needed for their tails, and the tails attract another kind of cell. So they work in different ways. Ultimately, we want to make lots of antibodies, and the antibodies help get rid of that foreign object. Now, there is something that didn't have a picture, known as activation of complement and inflammation. Complement and inflammation, if you haven't watched the video yet for chapter 14, these are second lines of defense. They are different types of proteins that can increase cell death, and inflammation can also, it's an advantage to have inflammation because it's activating lots of things wherever the infection is. So antibodies can increase both of those. Now, there are 5 different types of white blood cells. Like on my PowerPoint, I just listed 5. If you printed it, you can have just little areas to write it. But anything I'd want to go over is on this particular slide. This will be my last slide today. So 5 different types of antibodies. How are they different from each other? As I said before, the different antibodies, they do vary in their hands. Again, there are millions of different kinds of hands to recognize millions of different things, but the bodies stay the same, kind of. There really are 5 different kinds of bodies and how they like to arrange themselves. So I'm going to start in the middle. The IgM, Ig stands for immunoglobulin. It's another way to say antibody. So when they say antibody M, immunoglobulin M, it's an antibody. This is the first one that always shows up in the blood. So if you do get an infection and we activate those B cells, they become plasma cells, they start churning out antibodies, antibody M is always the very first one that we will see in our bloodstream. So it's considered the first responder. It's also the coolest one too, because it always hangs out with 5 antibodies together. So it's a pentamer. I always think it looks a little more like a snowflake. So it always has 5 bodies that are stuck together, which means it now has 10 arms on the outside of it, which means it's really good at agglutination, gluing and clumping things together. I mean, it's got 5 different hands. It can attach to 5 different bacteria, same kind of bacteria, but different ones. IgG is the most common antibody that's produced in the body. Reason why it's the most common, this is the one that's always produced the most with every reoccurring infection. So IgM is always the very first one that will show up the very first time you're exposed to something. But then every other time you're exposed to something, and you're exposed to lots of things all the time, you make lots of IgG. It's also the longest lasting, which makes it the most common. It is found in blood. That's the serum, the liquid portion of the blood. And it can cross the placenta, which means it does provide a developing infant some protection. Again, most things can't cross the blood barrier from the placenta, but a few things can. IgA is associated with all the secretions that come from your body. So your sweat has antibodies in it. Your tears have antibodies in it. Anything in your digestive tract has antibodies in it. Breast milk has antibodies in it. It's one of the top reasons why they encourage new mothers to breastfeed, is because whatever mom was ever exposed to, like ever, and however many years that she lived before that, she is making antibodies against it, which means that infant is now protected against anything. However, as soon as you stop breastfeeding, no antibodies, infant has got nothing. The IgE is only involved and seen at high numbers in allergies, if you ever have allergies, and a parasitic worm infection. It's the only time you really see these two particular, when you see this particular antibody. And the last one, the IgD, we find it hanging out next to the B cells in the B cell membrane, but we're not exactly sure what it does. There's still some things we don't completely know. They're working on it. Maybe by the next textbook edition, they'll have a better idea. So different kinds of antibodies found at different places and do a little bit different things.